Mark, no pressure. So yeah, I am from the northeast. I grew up in West Auckland. All my grandparents grew up within walking distance of where I was born, within walking distance of where I was born. So I'm very much a representative of the genetic makeup of this little part of the world, and in fact took part in the people of Great Britain for that reason. This is a picture of me that appeared. That's me standing over on the side there, holding an empty tube and looking at the guarantee for the fridge and trying to look intelligent. And that appeared in the medical school prospectus in Newcastle for many years. It was taken in 1971, just after I'd started there. And I think they put me in because I had long hair. I looked a bit like Andy Carroll back in those days. I, just at that point, decided to leave my medical course for a year to do a degree in genetics. The reason I did that was twofold. First of all, I went to Lumley Castle to a lecture. Very enthusiastic biology teacher, Mr. Mason, and his wife Mrs. Mason, who taught us, took us to listen to an academic from Oxford explaining the genetic code. Now, you've all heard of the double helix. Francis Crick and Jim Watson, who discovered that DNA is a double helix, and that's how we pass on our genetic material, by splitting that helix in half and making copies from it. But it took a mostly British bunch of scientists another 15 years or so to work out what the code was, to actually break the hieroglyphics. There are 4 letters involved, and those 4 letters are combined in sets of 3 to represent the building blocks of our body. And that's the actual code that was cracked in the 1960s. And there are the 20 amino acids plus 3 full stops made up from those 64 possible combinations. It's called a degenerate code because, in fact, you've got a lot more combinations of 3 letters, or 3 words of 3 letters from 4 letters. Than you need, if that makes sense. In other words, you can make 64 combinations of 3 out of 4, but you only need a dozen of them. So that's a problem I'll come back to in a minute. On the way back to school, our biology teacher came and sat next to me and asked me to explain it to him again. And I was really chuffed by that, because that's the first time I'd ever had one over on him. So I thought, oh, that's something in this genetics. The other reason I took a year out was because my dad was a businessman, he was an entrepreneur. And he used to, among other things, sell pit heaps and railway lines. And in fact, I ran my own business in Elton when I was 17, selling coal door to door. I used to take it from his pile of coal until he realised I was undercutting him and then stopped. But it made me aware of the concept of catching a wave and catching a growing edge. And it struck me that this discovery was going to make a huge difference in the world, and nobody in the medical school seemed to have heard of it. So basically in fact, this little thing, so if you take those 3 letters you end up with proline, but in fact any one of the last letters could give you a proline, so there are 4 different ways of doing that. And that's one of the problems that we have in understanding when we read people's DNA. This is Jim Watson, who is the patron, who, along with the late Francis Crick of the Centre for Life that we built in Newcastle and the Queen opened in 2000. And Jim still comes to visit regularly. He, among other things, set off the Genome Project in the 1980s, which was to sequence the human genome. And that was a huge multi-billion pound process. We think about $3 billion was spent on it, and about a quarter of it was done here in Britain. By 2000, when we opened our centre, it was costing about £100 million to sequence one person's whole genome. And in 2007, something quite remarkable happened. A discovery was made that you could actually sequence DNA in a different way, and the price started to fall. And it's now fallen to the point where you can sequence all 20,000 genes from a person for about 2,000 quid, and it's going down even faster. So we're actually heading for something that we thought was unimaginable a decade ago, the $1,000 genome. That's 600 quid in our money, and that's really quite remarkable, and it's going to transform our clinical practice. In fact, a team in Oxford have just invented, the Oxford Nanopore, invented a device that still actually doesn't really work, but it looks good. And it's called Minium, but that actually you stick it inside of your computer on a USB port, and for a few £100 you can sequence thousands of genes. It's got too high an error rate. Ours is going to be better, but I'll come back to that in a minute. But the point is that all sorts of headlines are now hitting the newspapers, and I'll speak about it some more later. So here's the problem that we're now running into. This is Jim Watson's analysis of all the mutations that were found in his genome when he was sequenced, the first human, along with Craig Bender, to be done in 2007. And I'm not going to take you through all the detail here. Look at the numbers on the bottom line. These are spelling differences between between him and the genome project that was done in 2000. 1.3 million, 1.4 million different spellings of individual bits of DNA in James Watson's genome. Almost all of which, in fact probably all of which, are irrelevant. That's because of that degenerate code, because you can change the spelling and get the same answer. And of course the genes themselves only make up about 2% of the total, so you can vary the spelling in between enormously. So our problem now is to deal with this huge burden of information. I was looking on the internet and I discovered in 2010 the world had its biggest traffic jam in China. It was 110 kilometres long and lasted for 11 days or something. And that's currently the world record, but that's actually nothing compared to the traffic jam that we're about to run into. You don't have to think for long to realise if, as is soon to be the case, I could sequence all of you, we're going to end up with maybe up to 2 or 3 million variants for each person to sort out. 1 or 2 of which in each of us might make a difference to our health. And I'm currently involved in an international exercise to try and pool our knowledge so that we can produce a sort of online database of which spellings matter and which ones don't. So that's sort of a long way from that lecture in Lumley Castle, but it's directly relevant to that first experience. Durham has actually figured very importantly in the other major research that I've been involved in, and it was about hereditary cancer. So in the late 80s I became involved with families where there was a gene running in the family that caused them to get cancer. And this is a family from Northumberland that I dealt with. And it's actually an important family because it was the first family in which a spelling mistake was found in what's called a mismatch repair gene. Ted there, who was both Ted and his sister are now deceased, but both of them had multiple cancers. In Ted's case, he had 3 colon cancers and 9 skin cancers. And we sent his DNA to some yeast geneticists in Boston because they were looking for DNA from families that had this problem. They had a hunch of what the cause was, and it turned out to be true. And the problem is that you inherit a spelling mistake in the spell checker in your DNA. So when you write on your computer now, you notice sometimes it will underline and point out a spelling mistake that you've made. The body's got exactly the same system of spell checkers that checks as the DNA is copied to make sure it's correctly copied. And if you have a problem with that spell checking system, if you inherit a problem, it makes you much more likely to get cancer because you make mistakes that can then lead to the cancer growing. So that was a huge breakthrough for us. And what was exciting and also interesting philosophically was that was actually shown in yeast. They used the genes from Ted, or the DNA from Ted, to look in yeast and find the gene that was going wrong in both yeast and in humans. We basically share exactly the same spell checker, more or less, as every other branch of the animal and plant kingdom, which is an interesting philosophical thought, sort of a Buddhist moment. If you ever sit and drink a pint of beer on your own, just think down there in the beer there's a yeast cell fixing its DNA, just like you are at the same moment. So Durham cropped up on this straight away because in fact, while we were involved in that research with the Boston team, we've been mapping this family as a result of a GP in Durham called Ted Knaggs, who's sitting at the front there, who's on my team. Most of the people in that photograph are actually from this very extended family, where there were 45 people who got bowel cancer in one extended pedigree. We call it a family, but actually most of them didn't know each other, and one particular chap from Durham didn't even know he was related to them at all. We found him, and he actually had a colon cancer the size of a cricket ball removed successfully and cured him, and he didn't know he had cancer at the time. So that was a huge breakthrough. It turned out that this family also had a spelling mistake in one of these mismatch repair genes. So it meant that back in the early 90s we were off and running with a whole new branch of genetics that I could get involved in. And the thing that they shared in common was this problem with repairing DNA. And the bits of DNA that are the hardest to copy are called microsatellites, the repetitive bits. If I gave you something to copy down on a piece of paper, if I put 9 or 10 ands in a row, you could very easily copy that wrong. You might put 8 ands or 10 ands instead of 9. And if you do that, you can screw up the whole gene. And so that's particularly true if you've got letters one at a time, because remember you're reading in sets of 3, so if you slip in one extra letter, everything moves down one and becomes gobbledygook. So these repetitive bits of DNA were the actual underlying problem. And basically these, as I said, the spell checker is this sort of collection of proteins that walk down your DNA and check that you've copied it correctly and repair any bits that are wrong.